Hi, my name is Allison Turk, and this is my video presentation for a scientific journal for Biology 2440. Alright, this article was published in 2012 by Han et al., and it is about the dysregulation of the RNA polymerase 1 during transcription and diseases. So RNA polymerase is a enzymatic protein that is responsible for transcription, so turning a DNA template into ribosomal RNA, and this is really essential for the cell to carry out daily functions so it can produce proper proteins. So in this article, Han discusses how some alterations within the DNA template strand or the binding of the subunits of the RNA polymerase to the DNA can cause some irregularities within the transcription process. So an example of that would be Treacher-Collins syndrome. This affects the gene TCOF1, and this affects the subunits of the polymerase and the binding site. So in order for polymerase to be active, a subunit called sigma has to bind to polymerase in order for it to carry out the transcription process. So in Treacher-Collins syndrome, the receptor, the sigma protein, isn't able to bind to polymerase, and this changes the structure of polymerase, making it not able to go through transcription, and cellular growth does not occur. This results in some craniofacial abnormalities and hearing loss, and is autosomal dominant. Another example would be a Bloom's syndrome. This occurs on gene BLM. So within this disease, instead of interacting with polymerase 1, the effects are on the promoter region of the DNA template strand. So the DNA template strand has a defect in the promoter region, causing the polymerase not to initiate transcription. This results in some mental disabilities, premature aging, skin atrophy, diabetes, and these people that have Bloom's syndrome usually don't live very long.